Living Environment Boot Camp Survival Guide. 100 Important Topics You Need to Know to Pass the Living Environment Regents Exam. Topic 1, Cells. 1. Homeostasis is the ability of an organism to maintain a stable internal balanced environment. Failure to maintain homeostasis can result in sickness or death. 2. Metabolism is the sum of all the chemical reactions that occur within the cells of an organism. 3. Organic molecules contain skeleton structures of carbon with hydrogen and oxygen. Organic molecule. Basic unit, Lego blocks. Foods found. Carbohydrates, starch. Glucose, simple sugars. Grains, vegetables, fruit. Proteins. Amino acid, 20 kinds. Meat, egg whites, beans. Lipids. Fatty acid and glycerin. Animal fats, nuts, oils. Nucleic acid. Nucleotide. Small amounts in all foods. 4. Organization of living things. Smallest cells, tissues, organs, organ systems, organism. Biggest 5. Organelles are the small parts that make up a cell. Each has at least one specific function. A. Vacuoles store waste and water, large in plant cells, small in animal cells. B. Ribosome, very small and is often represented by a dot, located on the ER or in cytoplasm. Ribosomes are where proteins are made, protein synthesis. C. Mitochondria the powerhouse of the cell where energy is made, the site of cellular respiration in both plant and animal cells. Formula for cellular respiration. Glucose plus oxygen, carbon dioxide plus water plus energy, ATP. D. Chloroplasts only in plant cells, where the process of photosynthesis occurs. Formula for photosynthesis. Sun's energy plus carbon dioxide plus water, glucose plus water plus oxygen. E. Nucleus is the control center of the cell and contains DNA, the program or code of life. F. Cytoplasm is the liquid media that fills the cell. G. Cell membrane. 1. Separates the contents of the cell from the outside environment. 2. Controls the transport of materials into and out of the cell, selective permeability. 2. Developed by Mr. Barone. 3. Cellular communication, recognizes and responds to chemical signals by using receptor molecules. 6. Passive transport or diffusion is the movement of molecules from areas of high concentration to areas of low concentration. No energy used. 7. Active transport is the moving a molecule from low concentration to a high concentration, uses energy in the form of ATP. 8. Human body systems. Name of system. What it does. Parts of the system. Organelles with the same function. Digestive. Breaks down food into nutrients and puts them in the bloodstream. Mouth, esophagus. Stomach, small. Intestine, large intestine, rectum. Vacuole and lysosome. Circulatory. Carries gases and nutrients throughout the body. Heart, arteries, veins, capillaries. Cytoplasm, ER, or Golgi. Respiratory. Exchanges carbon dioxide and oxygen. Lungs, alveoli. Diaphragm. Cell membrane. Excretory. Removes wastes from the blood and then from the body. Kidneys, bladder. Urethra. Cell membrane or vacuole. Nervous. Fast control of the functioning of all body systems. Brain, spinal cord, nerve cells. Nucleus. Endocrine. Slow control of the functioning of all body systems. Pituitary, thyroid. Adrenal, pancreas. Gonads. Nucleus. 3. Developed by Mr. Barone. Topic 2, Life functions and homeostasis. 9. Control, chemicals produced in the endocrine glands, hormones, and chemicals produced by nerve cells are primarily responsible for communication between cells. Receptor sites. 10. Respiration is the process used by all organisms to produce energy by using oxygen to burn sugar in order to release energy in the form of ATP. Organelle used in this process, mitochondria. 11. Digestion, breaking large molecules down into smaller molecules. 1. Proteins are broken down into amino acids. 2. Carbohydrates and starches are broken down into simple sugars. 3. Fats, lipids, are broken down to fatty acids and glycerol. 12. Transport involves the movement of materials inside the cell as well as the movement between parts of a multicellular organism. 13. Excretion, the removal of all waste produced by the cells of the body. 14. Synthesis is the making or building of large molecules from smaller ones. 15. Photosynthesis is the process of storing the energy from the sun in the chemical bonds of glucose, sugar, organelle used in this process, chloroplast. Formula for photosynthesis. Sun plus carbon dioxide plus water, enzymes glucose plus oxygen plus water. Where is photosynthesis carried out? Chloroplasts of producers, plants. 16. Cellular respiration occurs in the mitochondria of all organisms, both plants and animals. Formula for cellular respiration. Glucose plus oxygen, enzymes carbon dioxide plus water plus energy, ATP, 4. Developed by Mr. Barone. 17. Enzymes, special proteins that affect the rate of chemical reactions. Enzymes are catalysts used in digestion and synthesis. Enzyme reaction rates are affected by 1. Shape, lock and key model. If it is the wrong shape it will not work. 2. Temperature, each enzyme works best at a specific temperature. 3. pH, each enzyme works best at a specific pH. 18. Dynamic equilibrium is a steady state balance, homeostasis. 19. Negative feedback, controls hormone levels to maintain homeostasis. Analogy equals thermostat, as the temperature in your house goes up, the thermostat turns your heat off, as the temperature goes down, the thermostat turns on. This maintains a balance or a comfortable home. A good example is how our body regulates the amount of sugar in our blood. When glucose, sugar, levels are above normal, the pancreas secretes insulin. This hormone prompts glucose to move from the blood into body cells, resulting in a lower glucose level in the blood. Another hormone secreted by the pancreas, glucagon, works in the opposite way. When the glucose level in the blood is too low, this hormone prompts the release of glucose stored in the liver, which raises blood sugar level. Negative feedback. 5. Developed by Mr. Barone. 20. Our skin and circulation systems are the body's primary defense against disease-causing pathogens. Immunity, via white blood cells. 21. Surface receptor protein, a molecule found on the cell membrane that the immune system recognizes as either part of the body or an outside invader. Antigens are the receptor proteins on the membrane of pathogens, germs. 22. Antibodies are special proteins produced by the white blood cells that can be thought of as your body's army to fight diseases. 1. Antibodies have specific shapes that fit over specific antigens. 2. When an antibody fits into the antigen receptor on the pathogen, it blocks communication, preventing the germ from reproducing and making you sick. 23. Immunity is our body's ability to fight disease. Once you have been exposed to a specific virus, white blood cells remember the antigens and produce antibodies that prevent you from getting sick from the same virus for the second time. 24. Vaccination is composed of a weakened or dead virus that triggers our white blood cells to produce antibodies to fight a specific pathogen. 25. Diseases are caused pathogens, virus, bacterium, and fungus. 6. Developed by Mr. Barone. Topic 3. Reproduction. 26. Cell division. 2 types, mitosis and meiosis. Mitotic division. Meiotic division. Asexual reproduction. Sexual reproduction. 1. Cell division 1-2. 2 cell divisions 1-4. Parent cell divides equally to produce two identical daughter cells. Parent cell divides twice to produce either 4 sperm cells or, female, 3 polar bodies plus 1 egg, ovum. Genetic makeup identical to parent 2n, diploid hashtag. Genetic makeup 1/2 of the parent cell 1n, haploid hashtag. Function, to produce identical cells used for growth and repair. Function, to produce sex cells with 1/2 of the species chromosome number. Mitosis is used for growth, repair, and asexual reproduction. Meiosis occurs only in the male and female gonads and solely produces gametes. 27 gonads are the sex glands. Ovaries and testis. 28 gametes are sex cells that unite in fertilization to form a zygote. 29 zygote is a fertilized egg. 30 fertilization is when a male and female gamete unite. If the gametes each have 23 chromosomes, then what does their zygote have? 46. 31 differentiation is the process that transforms developing cells into specialized cells with different structures and functions. Tissues are specialized groups of cells. 7. Developed by Mr. Barone. 32 human female reproductive system. 1 vagina. 2 uterus is where the baby develops. 3 ovaries are the female gonads that produce the egg and important female hormones. The placenta is the organ that is attached to the baby via the umbilical cord and provides nutrients, oxygen to the fetus and removes waste through the process of diffusion. Mother and baby's blood does not mix. 4 oviducts or fallopian tubes are the place where fertilization occurs. Placenta. 33 human male reproductive system. 1 testis is the male gonads that produce sperm and male hormones like testosterone. 34 reproductive technology. 1 artificial insemination, using sperm from a donor. 2 amniocentesis, removing some of the cells from the amniotic fluid which protects the fetus and analyzing their DNA. 3 karyotype, is a visual map of chromosomes. Can be used to see if the fetus has any chromosomal problems like Down syndrome, 3 copies of chromosome number 21. 8. Developed by Mr. Barone. Topic 4, Genetics. 35 Cancer occurs when certain genetic mutations in a cell can result in uncontrolled cell division. Cancer can be caused either by heredity or environmental factors like radiation, chemicals, or virus. 36 Heredity is the passing of genetic information from one generation to the next through reproduction. 37 The hereditary information DNA is organized in the form of genes located in the nucleus of each cell. 38 Differences between asexual and sexual reproduction. Asexual reproduction. Sexual reproduction. Identical cells. 1 parent. Little genetic variation. X, amoeba, paramecium, fungi mitosis. Not identical cells. 2 parents. Much genetic variation. X, humans, plants. Meiosis and fertilization. 39 Identical genetic copies are known as clones. 40 DNA is a double-stranded helix polymer of nucleotides that contains the genetic code of the individual. 41 Nucleotide is the basic unit of DNA which is made of a phosphate, a sugar, and a base. 42 DNA bases are A, T, G, C. A is paired with T, G is paired with C. All teachers go crazy. 43 RNA is a single-stranded polymer that is produced by DNA. 1 There are 3 types, messenger RNA, transfer RNA, and ribosomal RNA. 2 Bases are A, U, G, C. Notice there is no T in RNA base code rule. 9. Developed by Mr. Barone. 44 How does DNA use RNA to make a protein? Protein synthesis. DNA is stuck in the nucleus, so it sends a messenger single-stranded mRNA to the ribosome where the ribosome reads the message and directs the tRNA, TRUX, to bring it amino acids. The ribosome then assembles the amino acids together in the correct order to make a protein. 45 Mutation is any alteration of the DNA sequence which changes the normal message carried by the gene. 1 Substitution, one base is put in the place of another. 2 Deletion, a base is left out. 3 Addition, a base is added. 4 Inversion, bases are switched. 46 Gene expression. An organism's environment can affect the way that some genes are expressed. Example: Himalayan rabbit, fur color is influenced by temperature. 47 Genetic engineering is a technology that humans use to alter the genetic instructions in organisms. 48 Gene splicing is cutting DNA and placing it into another organism. Example: insulin, putting the gene for insulin into bacteria, and the bacteria produces insulin for humans. 49 Restriction enzyme is an enzyme that cuts DNA in specific places and is an essential tool in gene splicing as well as in gel electrophoresis. DNA fingerprint from gelphoresis: just like a barcode, the more common the bars, the more common heritage or ancestry. 50 Selective breeding is a process of picking parents with favorable traits to produce those traits in the offspring. X. Domestic animals and hybridization of plants. 51 Species is a group of closely related organisms that share certain characteristics and can produce offspring capable of reproduction. 10. Developed by Mr. Barone. Topic 5. Evolution. 52 Evolution is the process by which organisms have changed over time from simple, single-celled, complex single-celled, complex, multicellular to complex organisms. 53 Natural selection. Nature selects those individuals who are best fit for the environment. 54 Overproduction. More offspring are produced than can survive. 55 Competition. The fight for limited resources. 56 Variation. Differences among organisms in a species. Sexually reproducing organisms have more variation than asexually reproducing organisms. 57 Adaptive value. Any trait that helps an organism survive and reproduce under a given set of environmental conditions. 58 Extinction is the disappearance of an entire species caused by a failure to adapt to a changing environment. Extinction occurs when the environment changes and the species cannot adapt. 59 Ancestry, cladograms or family tree. 11. Developed by Mr. Barone. Topic 6. Ecology. 60 Ecology is the study of how organisms interact with the living and non-living things. 61 Biotic factors, are living parts of the ecosystem, ex plants and animals. 62 Abiotic factors, non-living parts of the environment, rocks, air, pH, sunlight. 63 Niche is a species' role in its environment, its job and what it eats. 64 Population, all the organisms of a species that live in the same area. 65 Community, all the different populations in an area. 66 Biosphere, all of Earth's ecosystems. 67 Competition, is the struggle for resources among organisms. 68 Limiting factors are the living and non-living things in the environment that limit the size of populations. Examples: food, shelter, sun, space, oxygen, etc. 69 Carrying capacity is the maximum population, number or organisms of any species, that an ecosystem can support. 70 Predators kill and eat other organisms called prey. Graph of prey/predator relationship. 71 Feeding relationships. 1 Autotrophs, producers, make their own food by photosynthesis. 2 Heterotrophs, must eat something for food, consumers. 3 Herbivores, can only eat plants. 4 Carnivores, can only eat animals. 5 Omnivores, can eat plants and animals, all humans. 6 Consumers, same as heterotrophs. 7 Decomposers, break organisms down and return nutrients to the soil. They are the recyclers in the ecosystem. 8 Scavengers, eat dead organisms that they did not kill themselves. Example, vultures. 9 Parasites, live off of another organism, host, and do not kill them usually. The parasite 12. Developed by Mr. Barone. Benefits: the host is harmed. 10 producers, same as autotrophs. 72 Food chain is diagram of the linear feeding relationship of organisms in an ecosystem. Rules in drawing a food chain. 1. All food chains begin with a producer, autotroph or plant. 2. All food chains end with a decomposer. 3 arrows in a food chain show the direction of the energy flow. Grass, grasshopper, frog, snake, hawk, decomposer. 73 Food web is a diagram composed of many interlocking food chains. It is more accurate than a food chain because most organisms eat more than one type of food in their environment. 74 The sun is the primary source of life energy on the earth. 75 Energy pyramid is a diagram showing the energy available at each trophic level. The bottom layer, producers, has the greatest amount of energy, and the amount of energy decreases by 90% at each level as you move up. Though the pyramid. Where does the energy go? Into the environment. Lost as heat. Energy is not recycled in an ecosystem, it is transferred from one trophic level to the next. 76 Self-sustaining ecosystem requires 1. A constant source of energy, usually the sun. 2. A process to capture the energy and store it in organic molecules, photosynthesis. 3. A way to recycle elements back to the ecosystem. Decomposers. 77 Ecological succession is the orderly sequence of changes in the communities living in a given ecosystem over time. Pioneer organisms climax community. 13. Developed by Mr. Barone. EX1. Ecological succession from bedrock to a forest. Rock lichens and moss grasses, shrubs, small trees, hardwood trees. 2. Ecological succession in a pond. Pond. Plants and organisms die over time, sediment builds, pond gets shallow, swamp develops, grassy field, forest. 14. Developed by Mr. Barone. Topic 7, Human impact. 78 Material cycles. 1. Carbon/oxygen cycle, photosynthesis/respiration. 2. Water cycle, evaporation, condensation, precipitation. 3. Nitrogen cycle, N2, nitrogen fixing bacteria, plants, animals, waste. 79 Biodiversity is a measurement of the degree to which species vary within an ecosystem. The more species present in an ecosystem, the higher the biodiversity. As biodiversity increases, the stability of an ecosystem increases. 80 How man has affected biodiversity. 1. Cutting down trees, for wood. 2. Planting all of the same crop in an area, loss of biodiversity. 3. Removed vegetation for houses, parking lots, roads, etc. 4. Killed organisms and destroyed the food web, because all organisms are linked to one another in one way or another. 81 Renewable resources, resources that can replenish themselves if not abused, planting of trees. Non-renewable resources, resources that take a long time to replace or form, like coal, oil. 82 Preserving our resources. 1. Reduce, cut down on the amount used, smaller packaging. 2. Reuse, use it for another application, old tires as flower planters. 3. Recycle, can be used to make the product again, like pop bottles. 83 Pollution is a harmful change in the chemical makeup of the air, water, or soil. 84 Human activities and the loss of diversity. 1. Direct harvesting, the destruction or loss of a species by overhunting. 2. Land use, finding the best way to live in the environment, building around trees instead of cutting them down and planting new ones. 3. Habitat destruction, destroying a part of the natural environment. 4. Deforestation, removing forests for wood or clearing trees for farms. 5. Imported species, invasive species or exotic species, example, purple loosestrife, zebra mussels, dandelions. 15. Developed by Mr. Barone. 85 Impact of technology and industrialization. 1. Industrialization increases pollution of air and water, uses more energy, water, fossil, and nuclear fuels. 2. Water pollution from sewage, wastes from homes and factories and animal wastes. 3. Toxic wastes DDT. 4. Thermal pollution, heat pollution. 5. Human overpopulation. 6. Air pollution, burning fossil fuels. 1. Acid rain, sulfur and nitrogen compounds in air pollution dissolve in the moisture of the atmosphere to form acids, causing rain to have a low pH, acidic. Acid rain kills trees and destroys historical artifacts. pH scale, 14 basic, 7 acid, 0. 2. Global warming, an increase in the Earth's temperature caused by an increase in greenhouse gases. Greenhouse effect is caused by increasing amounts of CO2 in the atmosphere caused by the increasing use of fossil fuels for energy. 3. Ozone depletion, hole in ozone layer caused by use of fluorocarbons. Ozone acts as a sunblock for all organisms on the Earth. UV causes skin cancer. 16. Developed by Mr. Barone. Topic 8. Experimentation. 86. Independent variable is the one thing that I change to test my hypothesis. The effect of 4. Sunlight, on DV, plant growth. 87. Dependent variable is the thing that changes because of the 4. Data collected. 88. Control group is the group that is studied under the normal conditions. 89. Experimental group is the group that is identical to the control group with the one change or difference, the independent variable. 90. Organizing data. Where does the independent variable, 4, go on a data table? What about the dependent variable, dv? Independent variable. Graph on the x-axis. Dependent variable. Graph on the y-axis. Ex sunlight. Ex growth. Where does the independent variable go on a graph? What about the dependent variable? Y-axis. dv. X-axis. 4. 91. Labeling a graph. 1. Title the graph, the affect of underscore 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 on underscore 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 dot. 2. Label the x and y axis. 3. Determine a proper scale for x and y axis. 4. Set up a key or legend if necessary. 92. How can you make an experiment more valid? 1. Repeat the experiment. 2. Increase the number of specimens or trials. 3. Peer review. 17. Developed by Mr. Barone. 93. Parts of the microscope. 1. Eyepiece, the part that you look through, closest to the eye, usually 10x2. Objective lens, the magnifying part closest to the slide, high power equals usually 40x, low power equals usually 10x. 3. Fine adjustment knob, used to focus on low and high power. 4. Coarse adjustment knob, used to focus only on low power. 5. Stage, where the slide is placed. 6. Stage clips, hold the slide in place. 7. Diaphragm, controls the amount of light used. 94. How to calculate total magnification, if a microscope has a 10x eyepiece and 10x and 40x objectives. Total magnification on low power, 10x 10 equals 100x, it looks 100 times bigger than real life. Total magnification on high power, 10x 40 equals 400x, it looks 400 times bigger than real life. 95. If you looked at the letter under a microscope, what would it look like? E. 1. Mirror image and flipped upside down. 2. Increasing magnification reduces the field. Larger image but you see less of it. 3. Increasing the magnification reduces the amount of light. Field darkens. 96. How to make a wet mount slide. Put the cells on the center of a slide, put a drop of water with a dropper onto the cells, do not touch the cells, lower a cover slip slowly at an angle, to reduce the number of air bubbles. 97. How to put stain on a slide without lifting the cover slip. Put a drop of the stain onto the edge of the cover slip, Place a paper towel on the edge of the other side of the cover slip. The paper towel will pull the water from under the cover slip, and in turn pull the stain onto the cells. 98 Laboratory tests to know. 1. Paper chromatography is a laboratory technique that is used to separate different molecules from one another. Separation of compounds in a solution by size and color. 2. Gel electrophoresis, separation of DNA using electrical current by size. 3. Indicators for pH, glucose, starch, color or state change indicating the presence of a particular substance. 18. Developed by Mr. Barone. 99 Dichotomous keys are tools to help in the classification of organisms. 100 Know the 4 state-mandated labs, Part D. I. Making connections, the Clothespin Lab. A. Part A. 1. What you did, measured how exercise affected pulse rate. 2. What you learned, exercise increases pulse rate. B. Part A. 2. 1. What you did, squeezed a clothespin for 1 minute, then squeezed it again for another minute. 2. What you learned. If you squeezed more the second round, it may have been because your finger muscles were warmed up from increased circulation. If you squeezed less the second round, it may have been because your finger muscles were fatigued. C. Part B. 1. What you did, designed an experiment to test how exercise affects squeezing a clothespin. 2. What you learned, how to design an experiment, see pages 3 to 5. 2. Relationships and biodiversity, Botanicurus Lab. A. What you did, compared 4 species of plants, based on structural, physical, and molecular, chemical and genetic, traits. B. What you learned. 1 species that are related share similar traits. 2 different techniques, such as gel electrophoresis and paper chromatography, can be used to determine relationships between organisms. 3 endangered species should be protected because they may offer benefits to humans. 19. Developed by Mr. Barone. Gel electrophoresis, a technique used to show how species are related to one another. Restriction enzymes cut DNA into fragments, which are placed into a well in a gel plate. An electric current carries the DNA fragments through the gel, separating them according to size. Smaller pieces of DNA are carried farther from the well than larger pieces. This creates a pattern of bands which is unique for every organism. Related organisms will show similar banding patterns because their DNA has similar base sequences. 3 beaks of finches. A. What you did, played finches with different beak characteristics competing for food. B. What you learned. Different environmental conditions, food, favored different species of finch, beaks, allowing some to survive and reproduce, but not others. Survival of the fittest, natural selection, competition, and variation drive evolution. Species change over time. 20. Developed by Mr. Barone. 4 diffusion through a membrane. A. Part A. 1. What you did. Made a model cell using dialysis tubing. Put glucose and starch inside your cell. Put starch indicator, iodine, outside cell. 2. What you saw. Inside of cell turned black because iodine diffused into the cell. Because outside of the cell was not black, you know the starch did not diffuse through the membrane. Used blue glucose indicator, Benedict's solution, to see that glucose did diffuse through the membrane. 3. What you learned. Small molecules, glucose, iodine, can diffuse through a membrane on their own. Large molecule, starch, cannot diffuse through a membrane on their own. You can use indicators to identify the presence of specific substances. B. Part B. 1. What you did. Looked at red onion cells under the microscope. Added salt water to the onion cells. Added distilled, pure, water to the onion cells. 2. What you saw. Salt water caused the onion cells to shrivel and peel away from their cell walls. Distilled water causes the cells to swell back to normal. 3. What you learned. Salt water causes water to diffuse out of a cell. In pure, distilled, water, water will diffuse into a cell. 21. Developed by Mr. Barone.